So I'm going to be talking about some work I've been doing for my dissertation at Louisiana State University on gene expression in migrating tree swallows. And I think it's always important to start by defining exactly what I mean when I say migration, because migration has different meanings in different contexts. In this context, when I say migration, I mean a regular round-trip seasonal movement between a breeding and a non-breeding area, with the key point being that round-trip part. So migration is a round-trip journey, whereas dispersal is one way. Migration occurs throughout the avian tree of life. About half of all bird species exhibit some form of migration. So that's thousands of species of birds that migrate, and because of the mobility implied by migration in birds, there are also some important diversification implications. My specific research interests are in the evolution and genomic basis of migration, and for this talk I'm going to be discussing a project that looks at the genomic basis of migration in a group of New World swallows. First, we can ask how we actually go about uncovering the genomic basis of a trait, and there are a few tacks we can take. We can look at candidate genes, so selecting a list of loci that we think are relevant to that trait. We sequence those loci, we look for sequence variation that relates to variation in our trait of interest. We can also do comparative transcriptomics, so sequencing expressed genes and looking for differences in gene expression that correlate to variation in our trait of interest. Or we can do comparative genomics, so sequencing full genomes and then scanning those genomes looking for regions that again show variation that relates to variation in the trait that we're trying to study. Research so far into migration on birds has followed these approaches, so we're looking at differential gene expression, taking a candidate genes approach, much more recently doing genome scale analyses, but the fact remains that we still actually know very little about the genomic basis of migration in birds and very little about the genes that underlie migration. Additionally, to date, there hasn't been a large-scale comparative transcriptomic study of migration in a wild, free-living bird, and so that's the gap that I'm seeking to fill with this project. So the question that I'm asking is which genes are associated with migration in tree swallows, and I'm taking a comparative transcriptomics approach to answering that question. So asking which genes are differentially expressed at different points along the migratory trajectory, looking specifically at brain and liver tissue. This talk focuses on work in tree swallows, Tachycina bicolor, which breed throughout central to northern North America and then migrate south through the southern United States to their wintering areas in Mexico and the Caribbean. One fundamental problem that we face in gene expression studies is the differences in gene expression are obviously influenced by many factors beyond simply the trait that we're interested in. And this problem is compounded in free-living organisms because we are simply incapable of controlling the environment that our study animals experience. And this means that when we are trying to look at differential gene expression in free-living organisms, we have to try to collect our samples from animals that are experiencing environmental conditions that are as similar to each other as possible to try to control for some of that environmental variation. And this, as I'll explain, is what makes tree swallows a great system to look at differential gene expression and migration. So as tree swallows migrate south, they stop and form temporary roosts and stopover sites. One of these stopover sites is in Vacherie, Louisiana. So we're in Louisiana again, which is great. And you can see that the site is right on the Gulf Coast, and so it acts as a staging area for these birds to stop over and get ready to actually head on migration across the Gulf to their wintering areas. The roosts form in sugarcane fields along the Mississippi River. You can see that patchwork of sugarcane fields along the river in the satellite image to the right. And at their peak, these roosts hold hundreds of thousands of birds, and we know from geolocator data that these birds are coming from across the breeding range and that they all have they're exhibiting different migratory schedules. So if we go into that roost and sample a bunch of birds, we're going to get birds that are leaving at different times, arriving at different times, that are at different points along their migratory trajectory, exhibiting different migratory behaviors. So we can go in, we can take a sample that exhibits variation in migratory behavior, our trait of interest, but because we're catching them all at the same place and at the same time, we're controlling for that environmental variation that we're so worried about. So November of 2012, we went out and caught 30 birds in Vacherie. We caught them all at dusk on the same night, so we can catch them at the same place at the same time, controlling for environmental variation. As birds prepare to leave on migration, they lay down increasing amounts of subcutaneous fat until they're fat enough basically to take off and migrate. The birds that we caught in Vacherie fell into fat classes 2, 3, and 4 on a standard 0 to 5 scale. So we are catching a fair amount of variation in environmental behavior using fat as a proxy for readiness to migrate. I want to point out that I'm going to refer to fat class 2 as lean birds, but birds that are not on migration don't tend to carry any subcutaneous fat at all. So even fat class 2 is considered to be in migratory condition. extracted total RNA and then pooled that RNA into 8 brain pools and 8 liver pools, and I'm going to explain that pooling scheme, which you can see at the bottom. So again, 8 libraries of liver, 8 libraries of brain, libraries 1 and 2, and again, there's a brain library 1 and a liver library 1, a brain library 2 and a liver library 2. Those pools have the same individuals in them. So libraries 1 and 2 are my lean birds. These are the birds that are furthest away from migrating. They have the longest amount of time remaining before they continue their journey. Pools 3, 4, and 5 are the medium birds. So these are the birds that are a little bit closer to leaving on migration. And then libraries 6, 7, and 8 are the fat birds. So these birds are just about ready to leave. They're the closest to heading off across the Gulf. And so lean, medium, fat are my 3 experimental groups that I want to compare gene expression among. We then sequenced these 16 libraries and 6 lanes of Illumina, which brought us back 1.4 billion 100 base pair single end reads. We got at least 85 million reads per library. Then we did some basic quality control and adapter trimming on the raw data. And then we moved into working on actual transcriptome and gene expression analysis. So we got the data back a couple months ago. I've been working to assemble a good reference transcriptome to map reads to do differential gene expression analysis on. But in the meantime, we decided to start trying to work on a method to actually pick out patterns in gene expression without having to use a reference transcriptome to do kind of reference transcriptome-free gene expression analysis. So to do this, we wrote Perl scripts that scan through all 1.4 billion reads, not just a read right there, 100 nucleotide long read. And it looks for a 3 nucleotide long seed sequence. We arbitrarily chose TCG. This could be any sequence. It could actually be of any length as well. In this preliminary search, we're trying to reduce our data set down to a manageable level. So the downside of that is that we're also missing a lot of sequence. So we then extract all non-overlapping 20-formers from the data set that begin with that seed sequence. So in other words, we start reading through our read, we hit that, we find that seed sequence, and we then take the 21 base pairs that follow that seed sequence and output the resulting 20-former to a table. We then keep reading through the read. In this case, we don't find that seed sequence again, so we don't get any more sequence out of that read. In some cases, we do actually find a seed sequence again, so we can get up to 3 20-formers from 100 base pair long read. We then kept all the 20-formers that appeared in the data set more than 10 times. This gave us 11 million unique 20-formers, 865,000 of which occurred more than 10 times in the data. So this is a set that we're going to use for further analyses. We can then count how many times each of those 865,000 20-formers occurs in each of our 16 libraries, in each of our 8 brain libraries, and each of our liver libraries. So that's what our data table looks like, except that the actual table has 865,000 rows, of course. And we do actually pick up on some patterns. So basically, we look at that table, we can filter that table to look for patterns that we're interested in. And one of the first, so I'm going to talk about a few of those here. These are very preliminary results. And so one of the first things that comes out is a pattern of tissue specificity. So there are a lot of 20-formers that appear in one tissue, but not in the other. So these 20-formers appear in high numbers in, say, all of the brain libraries, but none of the liver libraries, and then the inverse. So there's about 7,700 brain-specific 20-formers. So again, 20-formers that appear at high levels in the brain and very little or not at all in the liver. And 1,300 liver-specific 20-formers with the opposite pattern. And so this is just that idea. This is 2 examples of that pattern shown graphically. And these bar plots I'm going to show are all going to have about the same format with the 20-former count on the y-axis, the 16 libraries along the x-axis. Again, that's 8 brain libraries and 8 liver libraries. The lean birds are white, the medium birds are gray, the fat birds are black. That's the 20-former sequence with just a black result below it. You can see on the top bar plot is a brain-specific 20-former. So again, that 20-former is showing up in all 8 brain libraries, but really not at all in the liver libraries. And then the inverse pattern is true with the liver-specific 20-former on the bottom. The next pattern we see is one that looks initially like a treatment association. So again, 20-former count on the y-axis, our 16 libraries right across the x-axis. Whites, lean, grays, medium. And what you can see here is that in the lean and the medium birds, in both the brain and the liver, we're seeing this 20-former coming up quite a bit, whereas it's not coming up at all in the fat birds. And so initially you might think that this is actually a gene expression difference relating to migratory condition, where birds that are further away from migrating are expressing this 20-former, whereas the fat birds that are closer are not. However, if we then scan through all of our 20-formers for sequences that differ from this one by only 1 nucleotide, slide, and that's the same bar plot, the black part of the bars. The top sequence is a sequence that differs by 1 nucleotide from the one from the previous slide, and that's the gray bars, and you can see that now that top sequence is being expressed in all of the libraries pretty constantly at pretty high levels. So it actually looks like the sequence polymorphism. So, and there's actually quite a few of these. There's a couple hundred 24mers that show treatment association. For a lot of these, we can find this pattern with the sequence polymorphism, suggesting there may actually be population structure in this species that correlates with migratory schedule. And then finally, we get patterns that actually do look like gene expression. And so bearing in mind that gene expression differences are probably going to be quantitative rather than qualitative, that's the pattern that we see. Again, 24mer count on the Y, our libraries across the X, white, lean, gray, medium, fat, black. And here you can see that this 24mer seems to steadily increase in expression as these birds get closer to the migratory departure date. And this is the pattern that I was really interested in seeing as a kind of differential gene expression difference. So in conclusion, we do seem to see loci that are differentially expressed among tree swallows with varying migratory schedules, and the presence of some sequence polymorphism suggests that there might actually be population structure in the species that correlates with migratory schedules. So some populations are migrating at a different schedule than other ones, and this is a pattern that we see in other birds. And so where do I go from here? First, I'm going to continue working on actually getting a transcriptome assembly together so we can do a traditional gene expression analysis. But we're also going to keep working with this reference-free analysis to see how far we can go, working on increasing the portion of data we're sampling by varying that seed sequence, and also working, of course, on doing formal statistical analyses so that we can actually compare the results from these two different analyses. And of course, there are a lot of people and organizations to thank, and if there's time, I'm happy to take any questions.
Some questions?
Is there any variation regarding where were they flying in flight?
Is there a variation depending on where they're going or where they're coming from?
Yeah, where they're going. So I suppose they go somewhere specific in this range?
They probably do, but it's impossible for us to know where based on, because we catch them in this gigantic roost and we have no idea where they come from or where they're going unless they're individually tagged, which in the geolocator study there were bit nuns that like killed them in the roost, and other than that, no, so we don't know. But that would be awesome to know.
Yeah, I may have missed this, but how did you control for batch effects between library effects and across sample sequences?
How do we control for what effects?
Batch effects due to different sequencing lanes or different library effects?
So we multiplexed all of the libraries across all of the lanes. Did that answer your question?
Yeah.
Yes.
Have you considered males and females in their analysis? Difference?
I'm sorry?
Males and females? Sex difference?
Yeah, so we did have a variety of males and females and different age classes, and each library I tried to balance those. The libraries have about the same composition of males and females and juveniles. But yeah, that is definitely a concern.
So how did you measure how fat? Like, how do you know how if a bird is fat or not? I've never.
There's a couple
Do you
ways.
weigh
So
it, or?
in live birds, it's called furcular fat. You can actually see it, and when they're really fat, it's like bulging. Like it's actually pretty gross. Also, because we euthanize these birds, we can also actually take the skin off and look
Right.
underneath the skin for fat. But traditionally, those fat stores are furcular fat.
Is the 24mers are loci that are linked to the actual gene sequence of, or are they actually found within coding regions of the genes that are expressed?
So at this point, we don't really know what these 24mers are. We just know that it's a 24mer that appears in the leaf.
Okay.
And that's one of the things we're doing is going back and pulling those reeds back out and fishing for those reeds with the 24mers and then trying to figure out what's going on with them.
So if there's a SNP between that distinguishes two of the 24mers, does that necessarily mean anything? Because if those are just linked to the actual loci that are behind what you're seeing, that SNP itself might not have an effect.
It might be a completely silent thing, it might be a sequencing error, or it might be something interesting. And again, like that's the downstream stuff that we're going to start trying to do, is figuring out some biological relevance to some of these patterns.
so they must not be seeking the error. They might also be error loss.
Yeah.
Also.